Welcome to Biology, My Passion. I am Soumya Harikrishna. We are learning the chapter Molecular Basis of Inheritance. Today, let us learn protein synthesis, otherwise called translation. What is protein synthesis? We know proteins are made up of amino acids. So, hundreds of amino acids are connected end to end with peptide bond to form long molecules of proteins. So, the polymerization of this amino acid to form a long polypeptide chain is called protein synthesis or translation. But we know it is made up of different types of amino acids. 20 different amino acids are involved in the process of protein synthesis. So, what will determine the sequence or the order in which amino acids have to be arranged? That is based on the information on mRNA. From where did this mRNA form? mRNA formed from the DNA by a process of transcription. So we know that DNA is the information center for making protein. In that, the codons are there. From DNA, what will form? mRNA will form. And we call this process as transcription. Based on the information on this mRNA, the proteins are formed. That is, each one is an amino acid. Like that, many amino acids are connected. So this is protein, which is a polypeptide chain, and here this process is called translation. So this is nothing but central dogma. We have done a detailed video on central dogma. We have also covered DNA replication and transcription. I request you to watch the structure of RNA, also the genetic code before we start with translation. What are the requirements of protein synthesis? So, protein synthesis to take place, we need amino acids first. Because we know proteins are made up of amino acids. So, amino acids are the raw materials for making proteins. Now, how many amino acids are needed? In living organisms, all proteins are made of almost 20 different types of amino acids. Now, where are they found? They are present in the cytoplasm as amino acid pool. Now, second requirement is DNA. We need to make protein, the same type of protein again and again. So, each time the protein should be similar to the one produced previously. So, the information should also be same each time. So, the DNA is more or less a stable molecule. We know, unlike RNA, DNA is very stable and its information is also stable. So, each time we can make the similar type of protein based on the information on DNA. But we know DNA has 4 types of nitrogen bases and 3 consecutive nitrogen base sequence make a codon or codes for an amino acid. But amino acid is coded by uracil, whereas DNA has thymine. So, from DNA, another intermediate is formed, that is mRNA. mRNA has triplet codons. Based on that, protein synthesis can happen or amino acids can be brought. Then, for this, another adapter molecule is required because there should be some molecule which can read the sequence on the mRNA and at the same time bring the correct amino acid to the site. Suppose the first one is methionine. So there should be someone who has to read the codon on the mRNA for methionine and bring the methionine from the amino acid pool. Okay, so RNA is like a adapter molecule doing these two. So TRNA means transfer RNA is the next important requirement for protein synthesis. And finally, we need a place to take place all this. That is called a protein factory. The place where protein is synthesized in a cell. Where is it? It is in an organelle called a ribosome. We know ribosomes are made up of mainly structural RNA and protein. They are non-membrane bound structures, right? They have only some RNA, ribosomal RNA and proteins associated with them. And in prokaryotes as well as eukaryotes, 2 subunits are there for ribosomes. A larger subunit and a smaller subunit. Are they always together? No. Once the protein synthesis starts, larger subunit and smaller subunit will come together. Then it becomes activated ribosome. Once the process ends, they again dissociate. Means they become inactive. Ribosome has got two subunits, a larger subunit and a smaller subunit. It has two binding sites. One binding site is there for the mRNA because it has to attach to the mRNA for the protein synthesis to happen. At the same time, there are three binding sites for the tRNA to attach. Which are the three binding sites? E site or exit site, P site and A site. So, P site is peptidyl tRNA site. The tRNA that contains the growing polypeptide chain will be attached here. I will explain that later. Then the next tRNA bringing the amino acid for attachment will come and attach here at the A site, that is amino acyl tRNA site. This E site is otherwise called exit site. That means the discharged tRNA will leave the ribosome from the exit site. Now let us explore the mechanism of protein synthesis step by step. We know that proteins are made up of amino acids. They are connected by peptide bond. Whenever there is a bond, what does that mean? It is capturing energy. So for formation of bond, peptide bond, energy is required. We know whenever we think about energy, we correlate it with ATP. So here the first step is to provide the energy to this amino acid for making the peptide bond later during protein synthesis and attach that amino acid with the cognitive tRNA. So let us see the first step of that is activation of amino acid. How do we activate amino acid? By providing it with energy. Energy from whom? Energy from ATP. So the first step is amino acid combines with the molecule of ATP in the presence of an enzyme, amino acyl tRNA synthetase, and magnesium ion is also required for this to form a complex. What is that complex? Amino acid AMP enzyme complex. So here ATP will break down to form AMP, adenosine triphosphate will become adenosine monophosphate. So pyrophosphate is released. Now we know that once ATP is broken down, energy is released. Now this energy is with whom? With this amino acid. So we can say that this amino acid is activated amino acid. Okay, so amino acid, in the first step, amino acid combines with the adenosine triphosphate to become amino acid AMP enzyme complex and pyrophosphate is released. So the energy thus released is stored in this amino acid for peptide bond formation later, and this process is catalyzed by an enzyme amino acyl tRNA synthetase. And we know this enzyme is very specific. So 20 amino acids, if we need to charge, 20 different types of these enzyme is also present in the cell. Once amino acid is activated, the next step is charging of tRNA. That is, this amino acid has to now bind to the cognitive tRNA. So amino acid AMP enzyme complex formed is now binding with the corresponding tRNA to form tRNA amino acid complex, which is now forming. That is, amino acid will bind with the tRNA. That's called a charged tRNA because the energy is there in the amino acid. If that energized amino acid is now bonding with the tRNA, right? tRNA has a 3-OH group. With this, the 3-COOH group of the amino acid will bind. Okay, that is carboxyl group of amino acid will bind with the 3-OH group of the tRNA and they join. So now the tRNA is charged because it got the energy through the amino acid. Now who is left out? AMP and enzyme are left out. This reaction is also catalyzed by the enzyme amino acyl tRNA synthetase. Suppose this is the amino acid. So first amino acid should have UAC because we know that the start codon is AUG. Corresponding codon is anticodon is UAC. So AUG codes for methionine. So now what happens? This amino acid methionine, which is already charged, is with the tRNA. So now this is the charged tRNA. This is the first step of protein synthesis. Now this charged tRNA will start moving towards the site of protein synthesis. Now the third step is activation of ribosome. So the smaller subunit, as I told, the inactive stage ribosome subunits are at different places. So initially what happens, the mRNA will come and bind with this smaller subunit. So suppose this is the mRNA, it's coming and binding with the smaller subunit with the start codon. Okay, now this is forming the initiation complex. For this, magnesium is very important, that you have to keep in mind. Okay, so this is initiation complex. Now who will come? The larger subunit will also come and join with this. The larger subunit also joined. Now who has to come? The tRNA. We know that the first site AUG is the start codon. So the next step is polypeptide formation. So polypeptide formation we can classify again into 3 steps. That is initiation, elongation and termination. That is starting, formation or extension of the polymer, then the ending of the process. So for that, first we will discuss initiation. So here now smaller subunit, mRNA, larger subunit ready. Now the next, so the first amino acid will also come. So the first tRNA is the initiator tRNA, which consists of the anticodon for AUG. What is the anticodon for AUG? UAC. And that codes for AUG codes for methionine. But there is a slight difference. As I mentioned in the genetic code video, in eukaryotes it codes for AUG codes for methionine, whereas in prokaryotes it codes for N-formyl methionine. So now this will come to the P site. P site is free. It will come to the P site and bind with it. So only the first one is allowed to come to P site directly. Hereafter rest all will be coming and binding with A site only. Do you know why? Because P site will already be occupied. Right. So the first step, this is called an initiator tRNA. Now comes with the methionine and binds at the P site. This is the initiation of translation or protein synthesis. Here this anticodon binds with the codon on the mRNA with the hydrogen bonds. For the formation of this initiation complex, who is helping? Initiation factors. So there are different proteins named initiation factors separately for prokaryotes as well as eukaryotes for the formation of initiation complex. Now we move on to the next step. What is that? Elongation. For elongation, what happens? Now we have to add more amino acids to methionine, that is the initiator amino acid, so that we can make a long chain of polypeptide. Another set of proteins called elongation factors help in this process. Again we can divide this second step, that is elongation, into 3 further steps. So the first step is codon recognition. So next one is GCA. So its anticodon is G for C, CGU. This will come and bind with the A site. Okay, A site with the CGU. So these two codons and anticodons, they bond together by hydrogen bonds because they are complementary to each other. They make a hydrogen bond. Now it codes for amino acid alanine. So now 2 amino acids are coming nearby on this ribosome. So these 2 amino acids now have to form a bond. Which bond? Peptide bond between them. So what happens is, this methionine and alanine both are charged, they are in close proximity. So the energy can be used for making a peptide bond. The amino acid methionine are on the P site and alanine on the A site are now making or getting connected through a peptide bond. Okay, so a peptide bond is formed between them. They are connected now. This process is catalyzed by an enzyme, peptidyl transferase. So peptidyl transferase is actually a ribozyme. Do you know what is a ribozyme? Ribozyme is actually RNA acting as enzyme, ribosomal RNA. We know the larger subunit is made up of, ribosome is made up of what? The structural RNA as well as proteins. So it's not the protein part, but the ribosomal part is acting as a catalyst. It's a segment of the large subunit of ribosome acting as catalyst for the formation of the peptide bond. So peptidyl transferase is the enzyme required for this process. Now, since these two are connected, this tRNA will detach from this methionine. Now the tRNA is removed from this. The second tRNA has now 2 amino acids attached to it. So the tRNA from the P site is now removed. What will happen? From P site, it will come to E site or the exit site. Is it charged now? No, it lost the charge. It is discharged. Charge is lost because there is no amino acid which was giving the charge. So, from this exit site, this can go out. tRNA on the A side has got now 2 amino acids connected to it by peptide bond. So, we call it as a dipeptide. The next process is translocation. So, since the linkage is lost, this tRNA, that is initiator tRNA, is now having no charge because it is separated from the amino acid, so charge is lost. So this uncharged tRNA now moves to the exit site. From this exit site, it will go to the cytoplasmic pool for bringing another amino acid from the amino acid pool later. By the time what happens, this A site from here, this one will shift to P site. This process is called a translocation, means transfer of location. From A site it is now shifted to P site, and for this process energy is required. So GTP is providing the energy required for this process. One thing we have to remember here is when this is moving, we know that this anticodon on the tRNA is bonded with the codon on the amino acid through hydrogen bonds. That is still there. So what happens when this is moving? It is pulling the mRNA also with this. So now that codon also is coming to this P site. Now this site is vacant, GUU. So we need another tRNA with the amino acid coding for it. So amino acid coded by GUU is valine. So now what happens? The next one is coming and binding here in the A site. Okay. So now alanine and valine are in close proximity. Can't they make a peptide bond? Now they will make a peptide bond. So now this process is taking place very fast. It takes only 0.1 second. Okay. So now what happens? Again it has to shift. The third one is now in the A site. Since alanine and valine form a peptide bond, now the linkage between the tRNA and the amino acid will be broken. This transfer RNA is now coming to the exit site. From exit site, it will move to the amino acid pool for bringing another one if the sequence is there for alanine. So now what is happening? The process is still continuing, the same process now. Now this on the A side, the tRNA is having a tripeptide there. 3 amino acids are there. So now from here, this will shift to P site. As I mentioned, it is pulling the ribosome also along with it. So now this side is vacant. Depending upon the type of amino acid sequence, the next amino acid will come here. So this is how the elongation of this is happening. And now polypeptide chain is also elongating. How long will it elongate? Till the process ends. But we know that the ribosomes are present on the surface of endoplasmic reticulum. So actually here there is a groove in the middle of this larger subunit. Through that groove, this newly formed polypeptide will be going to the endoplasmic reticulum. The newly formed polypeptide chain is called a nascent polypeptide. Now let us see how the process is terminating. So this is continuing. Many codons are here, and as per that the polypeptide chain also elongated. Finally, there will come a stop codon. We know there are 3 stop codons or termination codons. Which are they? UAA, UAG or UGA. I usually remember like UA, UAG and UGA. Okay. So UAG is here, UAG. Now we know that the initiator codon, that is AUG, is starting codon and also it codes for amino acid methionine. So initiator tRNA could read it. But for stop codons, they don't code for any amino acid. So no tRNA will come to A site and read it because they don't code for any amino acid. So the process is stopping. But there should be an end to any process, right? So here another protein called a releasing factor. There are different types in prokaryotes, RF1, RF2 and RF3. They come and join here at the A site. Now the releasing factor is coming here. once the releasing factor comes here, what will it do? It will add a water molecule to this end, okay, which is connected to P side. So as a result, this will cleave out of this tRNA. From this tRNA, this is separated. Now the polypeptide is completely separated. Okay, so the protein is released. It will go to the endoplasmic reticulum. Now the tRNA also will come to exit side. From there it can go out. Now what will happen? The two subunits of ribosome also will dissociate. They are inactivated. So now the newly formed polypeptide, it is a very long stretch. I made only 3 just to make you understand. So this long one will now, it is actually a linear structure. So linear structure is forming the primary structure of protein. You learned about the conformation of protein in class 11 biomolecule chapter, right? And if it is coiling, then it is called a beta structure, the secondary structure of protein is there, or it may also undergo coiling to form tertiary structure. All these within the same polypeptide, it folds to form secondary or tertiary structure. But sometimes it may join with the other polypeptide to form quaternary structure also. Also learned about polysomes or polyribosome. But here we saw only one ribosome doing the function. Actually, one protein is completely translated using one ribosome. Then what is the significance of polysome or polyribosome? Let us see that. Imagine the first ribosome we discussed has now reached here, almost moving towards the 3' end. Meanwhile, the polypeptide is becoming longer. Meanwhile, a new one will come and attach here at the initiator side and start forming the same type of protein. Protein cannot be different here. mRNA has information for a particular protein, right? But that protein we need more molecules. So, another molecule of the same protein will be synthesized here, and this will reach once here, the next one will join, then next one will join. So it is making a chain of ribosome called a polyribosome. So these are the different steps in translation or protein synthesis. As I mentioned, first the charging will happen, then the cognitive tRNA will bind with the amino acid, then the tRNA becomes charged. The ribosomes unit will come together, mRNA will come and bind to it, and the tRNA also, initiator tRNA also will come and bind, forming an initiator complex. Then goes to the stage of elongation, where the peptide bond is formed between adjacent amino acids which are there in the P site and A site. New amino acids will be added based on the information on the mRNA. So this process will continue till it encounters a stop codon. Once stop codon is found, the release factor will bind and thereby terminating the process. Cleave the polypeptide from the tRNA by adding a molecule of water. Hope you understood translation very well. If you like my videos, please like, share and subscribe to my channel, Biology My Passion.